Hello, ladies and gentlemen from Hamburg in these very special and challenging times. We hope you're all doing well and welcome you to today's live broadcast on how to get the best out of your cell-based assays. Our webinar will be presented to you by my lovely colleague, Dr. Jessica Wagener, and by Dr. Erik Bonke from our cooperation partner, Promega. Both presenters are application specialists focusing on cell handling in their respective companies, and I will give you a short introduction about both in a minute. My name is Christian Haberland, Marketing Manager for Instruments and Systems at Eppendorf, and I will be your moderator for today's webinar. At the end of the presentation, I would like to give you a quick overview about solutions that might be helpful for you to improve your daily cell culture work. Now, a little bit of technical information that is necessary for this webinar. Before we begin, I would like to encourage you all to get the most out of this webinar by submitting as many questions as you want at any time you want during the presentation. Just simply type them into the Ask a Question box on the left and click Send. We will answer as many questions as we have time for at the end of this webinar. If you have any trouble to see or hear the presentation, click on the Support tab at the top right or report your problem by clicking on the Ask a Question box on the left, and we will help you. Continuing education credits can be obtained by clicking on the tab at the top right and just following the process. Just one comment, if you want to stay updated on all upcoming cell culture webinars, just simply click on the Inside Cell Culture tab that you can see on the top. Just type in your name and your email address to receive a monthly newsletter that is dedicated to cell culture experts with helpful videos, posters, tips and tricks for your daily work and more. And you can also find an example of the newsletter on the registration page. Now, let me shortly introduce the presenters of your webinar today. Dr. Jessica Wagener from Eppendorf and Dr. Erik Bonke from Promega both hold a PhD in biology with a focus on and many years of practical and theoretical experience in cell culture. Both are amongst the most experienced application specialists focusing on cell handling in their respective companies and frequently share their knowledge in workshops and seminars around the world. You can find a complete biography of both speakers by just clicking the Biography tab at the top. Today, we are happy that Jessica and Erik are going to share their knowledge with us. So, enough of the talking from my side. Let's get started. Jessica and Erik, the stage is yours.
Thank you, Christian, for the introduction. And welcome everyone also from my side, and thank you for joining our webinar today. I want to start with looking at the contents of today's presentation. First, I will talk about the general aspects of cell culture routine and how to increase reproducibility of your data when preparing the cells for an assay. And then Erik will take over the next part and go into detail about optimizing assay setups. In the end, I will touch the topic of how to avoid cryptic contamination that can influence your assay results. Here is an overview of the cell culture workflow. So independent of the specific cell type you're working with, these steps are part of the lab routine. For this webinar, Eppendorf and Promega are joining forces to support you in optimizing your cell-based assay results. We will cover the general aspects like optimal seeding techniques and cultivation conditions and go into the details of different modes of detection and assay setup. But before we proceed, here comes our first survey. I'm interested in the cell types that you are working with. So please select all that applies. So what kind of cell types are you using? Continuous cell lines, stem cells, or other primary cells? We will wait a couple of seconds and wait until the answers are coming in. So I will read the question again. What kind of cells are you using? Continuous cell lines, stem cells, or other primary cells? Okay, and I can see the votes are coming in right now. I think we will wait a second more until we share the results with you. Okay, so I think we can close the poll now and share the results. So I hope that you can all see the columns now. So we see that the majority of you is working with continuous cell lines, almost 80%, 18% of you is working with stem cells, and about 45% of you is working with other primary cells. Okay, so thank you for participating. So no matter which cell types you are working with, there are some aspects that can be considered to increase the reproducibility and consistency of your cell-based assays. And this is what we are looking at in the next chapter. So let's have a closer look at this first experimental step that is so important, the seeding of cells. The precondition for getting comparable results in an assay is to seed out the same number of cells for every negative control, positive control, and for every experimental replicate, of course. Variations in seeding cell numbers and the formation of air bubbles during seeding will not only increase standard deviations, it will also make your results less reliable. When you fill, for instance, a multi-well plate with cells, it will take some time until you have filled all the wells. The longer the seeding process, the higher is the variation in cell number from one well to the other. As you can see in the image on the upper right, the cells sink towards the bottom of the tube with time, and this sedimentation goes quite fast, within minutes. Therefore, you need to resuspend or mix the cells to swirl them up in between to get an equal number of cells in every well. Saying that, we should keep in mind that cell culture media tend to foam due to the high protein content of supplemented serum. Air bubbles can hinder cell attachment. In the lower image here, you can see how an air bubble hinders the cells nearby to adhere properly to the substrate. So you should avoid harsh and fast pipetting to reduce the formation of air bubbles and also to reduce the shear forces which further stress the cells. In fact, there are a lot of aspects about pipetting that are often underestimated. Pipetting can have a big impact on cell culture experiments. Not only the type of liquid influences the pipetting result, also the pipetting technique plays a role. So when aspirating the liquid, hold the pipette vertical as seen here on the left image and immerse the tip only slightly and try to keep a constant immersion depth. If you follow this technique, you will improve the precision of the volume that you are pipetting. Now let's have a closer look on how pipetting can affect a cell-based assay. In this graph here, you see the results of a cell viability assay. Each column represents one row of a 96 well plates. The aim was to seed an identical number of cells in each well. 2 rows were left out for blanks. The cells were seeded with a manual 8-channel pipette from a reservoir, and after 24 hours, the number of viable cells was determined using a colorimetric assay. In rows 1 to 5, you see varying cell numbers in the wells and high standard deviations. Here, the user did not mix the cell suspension between pipetting each row. So the cells sedimented in the reservoir with time, and in addition, the pipette was held in different angles and heights during aspiration. In rows 6 to 10, you see the results with mixing the remaining cells before pipetting each row, aspirating at constant height and in upright position. Taken together, these data show that the pipetting technique has a big influence on the results of an assay. Besides the technique, the other thing that can affect results of an assay are the so-called plate position effects. If you work with a 96 well plate, the usual volume you use is 100 to 200 microliters per well. Especially during longer incubation periods, evaporation can become a critical factor with a small volume, especially in the outer wells. This is a phenomenon that we call the edge effect. So the outer wells of the plates should not be used for analysis because the evaporation can lead to different cellular responses. In the CO2 incubator, the conditions for the cells should be as stable as possible during incubation. Every time the door of the incubator is opened, the atmosphere inside is disturbed. It is a good advice to reduce the traffic in and out of the incubator as much as possible. Only open the door when necessary and close it as soon as possible. Some incubators show temperature overshoots to reduce the recovery time after a door opening. This can harm the cells, so your incubator should have a quick recovery of CO2 and especially temperature without any overshoot, as seen here on the upper right graph. Especially when you work with very sensitive cells or when you work in a lab where many people are sharing an incubator, it can be a good idea to have an incubator with segmented inner doors, like you see it here on the lower right picture. The small opening area helps to keep the atmosphere inside stable. In some cases, it can be a good idea to even have separate incubators for maintenance cultures and running experiments. So you seed the cells for an assay, do the treatment, manipulation, or whatever you want to do, and put the cells into a separate incubator for the time of the experiment. Your colleague who has to split the cells or change the medium uses the other incubator, and thus your experiment is not disturbed so much by fluctuations of the inner atmosphere. Temperature may be the most critical parameter in an incubator because deviations from the optimal temperature can be harmful for the cells. Here on the right, you see results of on the left, you see the results of a temperature measurement in different incubators according to a Dean standard. 27 different spots were measured inside the incubators, and as you see, some devices show quite high deviations from the optimum, as indicated here by the darker blue and red dots. Shown on the right image is an example of how cells can react to non-optimal temperature. In this case, we incubated human pluripotent stem cells at 37 and 38 degrees, and you can see that the 1 degree difference results in quite obvious change in morphology of the cell type. This stress situation might not be so obvious for every cell type, but as you can imagine, even less obvious cellular responses can influence your assay results. Monitoring cultivation parameters and documenting performance of your incubator are measures to establish long-term reproducibility of the experiments in your lab. Here on the lower right, you see a screenshot of the menu of the Eppendorf Cell Expert CO2 incubator. The red arrow indicates a door opening, and you see that the curves of CO2 and temperature are dropping. You can simply check for door openings or track back the performance data to see what might have influenced your assay this time. So before you start troubleshooting the assay itself, it might be worth looking into the performance of your incubator. Another stress factor for cells are vibrations. Here are two simple examples. On the left, you see cells that are suffering from frequent door openings, and these door openings cause uneven adhesion of the cells to the substrate they are growing on. On the right, it is shown what happens when you move vessels with freshly seeded cells. So, for example, you want to put your Petri dishes into a crowded incubator and need to push other vessels aside to make space on the shelf. This manual moving can disturb even cell adhesion and distribution. External vibration sources in the lab include running centrifuges, fridges, or air conditioning. Some incubators have a fan inside, which is obviously one of the most critical sources of vibration because the cells are placed more or less directly next to the fan. So whenever possible, incubators without internal fan should be preferred, especially for sensitive cells or sensitive experiments. Let's have a closer look at the cells in culture. So no matter what you do with a cell culture, it is important to determine the right time point for a particular cell type. In this graphic here, you see a typical growth curve of cells in culture. It is important to not let the cells overgrow until they are in their stationary phase and do not proliferate anymore. The optimal time point is when the cells are in their exponential growth phase, also called log phase. And this is when they are between 70 and 80% confluent. A growth curve provides information about behavior of a certain cell type during cultivation and thus facilitates the assessment whether any biological response can be attributed to the effect of a tested factor or whether it may reflect an imbalance within the cell culture system. It has often been described that the passage number influences cellular behavior. The passage number is a record of the number of times a cell culture has been subcultivated. There are numerous studies giving evidence that the passage number affects a cell line's characteristics over time. It has been described that cell lines at high passage numbers experience alteration in morphology, response to stimuli, growth rates, protein expression, transfection efficiency, and more. So looking at all these passage number effects in different cell types described in the literature, the basic question one can think of is how many passages are too many? And before we go on with the presentation, I want to hand over this question to you and start our next survey. What do you think? How many passages can a cell line be cultured for? So the answers you can click on are not more than 20 passages, 20 to 60 passages, up to 100 passages, or the passage number has no influence on the cells. So what do you think? How many passages can a cell line be cultured for? Not more than 20, 20 to 60, up to 100 passages, or the passage number has no influence on the cells? And again, I will wait a couple of seconds and give you time to answer. So I see the answers are slowly coming in. The columns are still changing, so I wait a couple of seconds more. Okay, and I think we can share now the results with you. Thank you for answering. Okay, I hope you see the results now. So around 60% says more than 20 passages. Around 34% says 20 to 60 passages is okay. Only 5% uses their cells up to 100 passages, and only 1% says the passage number has no influence on the cells. Okay, so thank you again for participating. I will go back to the presentation. All right, so in general, we can say keep the passage number as low as possible. Continuous cell lines like cancer cell lines basically have an unlimited lifespan, but although they can proliferate indefinitely, their genotype and phenotype can change over time. A lot of the well-established and well-characterized cell lines that are used in laboratories worldwide have been isolated from their original tissues decades ago. When you work with such an old continuous cell line, it is most likely not possible to obtain early passages from this cell line, though. But you have to set a starting point as a reference to be able to obtain reliable results with this cell line. First of all, the starting point should be cells of high quality. The best way to ensure this high quality is obtaining cells from established cell banks. Here you get well-documented, authenticated cell lines which serve as the best reference culture for your experiments. By establishing a reference cell stock, you have a constant supply of early passages from your cell lines. Now you can determine the safe passage number for that particular cell line of interest. By establishing certain baselines, for example, and test the cells for the presence and expression level of a certain protein, monitoring the proliferation rate over time, and keep an eye on any changes in cellular morphology. By getting to know your cells, you will get an idea of whether and how the cells change in relation to prolonged passaging. And maybe very simple advice, do not keep cells in culture that are not actively used for experiments. So don't passage continuously, but rather establish new cells from frozen stock if required. And now I would like to hand over to Erik from Promega, who will go into detail about optimizing the setup of your cell-based assay.
Thank you, Jessica. And thanks everyone for joining this webinar. So now that we've learned a lot about how to ideally handle cells, in this next section of the webinar, I would like to focus on how to use these cells in an experimental setting, a so-called cell-based assay. And primarily, I'd like to touch on different key factors known to significantly contribute to the outcome of such an assay. I will start by introducing the various detection modes available and highlight their principles and features. And then also related to this, I'd like to spend some time talking about how to choose the appropriate microtiter plate. We will then continue to talk about how the actual design of an assay, the cells themselves, as well as the readouts, so the marker that you choose to determine with your assay, and finally also the assay timing can affect your data. So let's have a brief look at the different detection modes available. The first option you have is an absorbance-based assay. Here you have an external light source that sends light through a sample, and what you basically determine is a decrease in light intensity due to partial absorbance of light by components within the sample. In general, one can state that these are the least sensitive assays. They suffer from a low signal to background ratio and a low dynamic range. These parameters are kind of improved in the second option. You have a fluorescence-based assay. Here again, we have an external light source that generates light of a distinct wavelength, and this light excites the fluorophore within the sample, and what you determine is the emission of light at a longer wavelength. What I would like to convey is that you will always yield the highest sensitivity with the third option, the bioluminescence-based assay. Here you have a signal generated directly within your sample in a continuously occurring chemical reaction, so there is no need for that external light source, and this is associated with a considerably lower background. As when you compare it, for example, to a fluorescence-based approach, where background can make up a large portion of your signal due to a phenomenon known as autofluorescence. Although at a first glance, the absorbance-based assays appear to be the least expensive ones, a major drawback is that usually they are not compatible with multiplexing. However, a fluorescence and a bioluminescence-based assay can be ideally combined in such a multiplexing approach. Multiplexing means that you determine two different parameters using one and the same sample. So in other words, you sequentially run two assays on the same sample. In order to do this, of course, these assays have to fulfill certain requirements. They need to be biologically and chemically compatible, and also they need to generate signals that you can easily distinguish. And last but not least, the higher end volume must fit with the microtiter plate format you use. Multiplexing is associated with several advantages. First of all, you have these two data sets generated with the same sample, so this also means that you maintain the same variables. But also, if you think of cost, this approach is beneficial because you can save sample material and also your consumables. But probably the most important benefit is that you can very easily normalize your data, and you can also verify it because you have these two separate data sets that can support each other. No matter what detection mode you choose, it is absolutely crucial to pick the appropriate microtiter plate. For any cell-based assay, you would want that plate to be a sterile plate, and it should have a lid. If you work with adherent cells, it should also be TC or tissue culture treated. You can then choose from clear, black, and white plates. The black and white ones are also available in different formats, a solid and a clear bottom format. The clear bottom format has the obvious advantage that you can look at your cells through the microscope. The disadvantage, however, is that you will lose signal to some extent. You have a higher well-to-well crosstalk, and also this format appears to be more expensive. For an absorbance-based assay, you would go for the clear plate, and depending on what region of the spectrum you're interested in, you can choose from different materials. The most important thing is that you must never use that clear plate for a luminescence-based assay. And I think that's conceivable if you think of that continuous light production that goes on in the wells of your plate. And in a clear plate, that light could go sheerly anywhere, so you could not track it down to a distinct well. The primary choice for a fluorescence-based assay is the black plate. Here, a black pigment is added to the plastic in order to decrease the autofluorescence of the plastic, so the background caused by the plate itself. And finally, the primary choice for a luminescence-based assay is the white plate. Here, a brightener is added to the plastic, which improves the reflective properties of these plates, so a maximum of your signal will actually be reflected towards the detector. And also in a luminescence-based assay setup, this decreases the well-to-well crosstalk. If you work with white plates, you should be familiar with phosphorescence because white plates can absorb and re-emit photons. And this is why we generally recommend you to store these plates in a dark space. And if you pipette them, to do this in a dimmed atmosphere, or if you insert them into your plate reader, leave them until you perform your measurement for a few minutes because that dark incubation allows the phosphorescent signal to disappear. What I also would like to highlight is that there are different qualities when it comes to microtiter plates. And this we showed here by a very simple experiment where we used two 96-well plates, two white plates from two different vendors. And what we did is we performed a serial dilution of a luciferase in the first column of the plate. The rest of the plate was filled with PBS. What you can nicely see is that in plate A, the signal generated within that first column of the plate almost travels all the way through the plate, while in plate B, it is mainly restricted to the site of its origin. And also the absolute signal intensities appear to be more higher for plate B than for plate A. So this suggests that the reflective properties of plate B are higher, presumably because a higher amount of that brightener has been added during the manufacturing process. In any case, plate B has the properties that you would want a plate to have, so a high signal output and a low crosstalk. Whenever you set up the cell-based assay yourself, you should always try to minimize the total number of steps required to perform the assay, because every step is a potential source of error and can increase the variability of your data. And this is exactly why Promega assays follow this common principle, the so-called admix and measure principle. This means we always have a single reagent that we only need to add to the cells, we briefly mix, and then we can read our signal. We also say our assays are homogeneous because we only have these addition steps and there is no further sample processing such as washing of cells or removal of supernatants required. And this is also what makes these assays HTS compatible and also what makes them easy to automate. But the fewer pipetting steps also make it easier for the scientists to actually perform the assays, and that makes them also time-saving. And last but not least, as I already mentioned, this very much improves the quality of your data. A nice example where we took the streamlining of an assay workflow to extremes are the so-called lumid immunoassays. This is probably one of the newest Promega technologies. And it is a technology that serves as an alternative to common immunoassays such as the traditional ELISA approach. I'm pretty sure everyone is familiar with a conventional ELISA workflow as it is depicted here. We have a very heterogeneous multi-step process that involves all these tedious washing and incubation steps and which can take you up to 2 days in time. With the lumid immunoassays, we were able to eliminate the majority of these steps. So we, for example, removed the coating and the blocking steps because we work in solution. We removed the washing steps and we combined the separate antibody addition steps into a single addition step. So by very much streamlining that procedure, we could eliminate a number of variability sources. And the nice benefit is that also these assays can be easier accomplished and faster accomplished, and it only takes 60 to 90 minutes. Another important factor associated with variability of data that is directly related to your experimental layout or assay design is the so-called edge effect that we've already heard of, and which mainly affects the outer walls of such a microtiter plate. And it is actually an effect that is apparent at two different levels. First of all, we have a higher evaporation in the outer walls than in the inner walls, and this leads to a concentration of salts and solutes within your medium. And this concentration can alter the growth and the viability of your cells. So what you actually end up with is a gradient of cell number across your plate. A good way to tackle this problem is to either use specialized plates such as Eppendorf offers, which have a moat around the outer wells that you can fill with liquid to insulate these wells. Or if these are not available, you can also fill the outer wells of such a plate as well as maybe also the interwell spaces with pre-equilibrated sterile liquid. The downside, of course, is that this decreases the number of samples that you can assay per plate. second level that this effect becomes apparent on is a temperature effect, because you can imagine that the outer wells will respond more sensitive towards changes in temperature than the inner wells. A scenario where we see changes in temperature frequently is the shuttling of the plate between the incubator and the reading instrument. So it is advisable to hurry up when you need to shuttle, or to avoid shuttling by leaving the plate in the reading instrument and by using this instrument as an incubator. In any case, you should pre-equilibrate all the reagent as well as the reading instrument to the temperature that you actually would like to run the measurement at. Also important for the reproducibility of your data is the seeding density of your cells. And while it is pretty conceivable that if you seed cells at different densities in microtiter plates, and if you treat them with a given dose of a cytotoxic compound, that these cells behave differently, it is not as obvious that also the seeding density in your stock culture flask can affect the outcome of your experiment. And this is shown in this experiment where we seeded cells in stock culture flasks at different densities, low, medium, and high. We harvested these cells and we seeded them at an equal density in a microtiter plate. The cells were then treated with a cytotoxic compound, and two parameters, ATP content and caspase 3,7 activation, were determined. What you can nicely see is that there's a clear difference in how these cells respond towards the treatment with vinblastine, and in fact the cells derived from the low-density stock culture flask appear to respond more sensitive than the others. So this is why it is really advisable to agree on different standard operating procedures in your cell culture lab, so that you make sure to always work with fixed time points when you harvest or seed cells, or fixed cell numbers, simply to reduce the risk for this variability source. Another variability source also directly related to your cells is their metabolic states. In these experiments here, we assayed different metabolic parameters using our energy metabolism assays. And here you can see the consumption of glucose and glutamine, which are the two substrates most commonly added to cell culture media. What you see is that within a time frame of 2 days, these substrates are mainly completely consumed. And of course, at that time point, when substrates become limiting, the cells need to adapt. They will undergo a metabolic switch in order to stay alive, and it is conceivable that these cells might behave differently in an assay than cells exposed to excess substrate. Going along with the decline in the substrate levels is a buildup of metabolic products such as lactate and glutamate. And the buildup of lactate is what you can visually easily recognize by a change of the medium color from red to yellow. And this acidification of media is a clear stress factor to your cells. And it can change their cell growth, but also the viability of the cells. So it might be a bad idea to culture your cells over the weekend, and on Monday, if you discover a flask like this, to use these cells for an assay. But also a shift in pH in the other direction in the basic region can be problematic. And this happens frequently if you use a bicarbonate buffered media, which most media are, and you leave your media bottle in an open state under the bench for a while, because then due to the low atmospheric CO2, your media will turn basic, it will turn pink. If you add that to your cells, it can have the same consequence. So you see, the viability is a really key factor for data reproducibility, and you should establish a cell culture routine that maintains a high viable cell culture. Cell viability assays can aid you in doing so. They can help you to compare, for example, different media compositions, FCS batches, or you can determine the effect of different centrifugation times and forces or trypsinization times. Common biomarkers for viability are the reduction equivalents such as NADH, ATP content and also DNA content, which changes upon division of cells. Then people look at membrane potential, or they determine the activity of certain enzymes. Instead of measuring viability, you can also measure cytotoxicity. Common markers for cytotoxicity are a loss of membrane integrity and the therewith associated release of cytosolic factors such as lactate dehydrogenase. People measure the activity of certain proteases or changes in membrane composition. And finally, also a decline in ATP and NADH content is indicative for a cytotoxic event. Importantly, depending on what marker you choose, you can then also make a statement on the mechanism that your cells have died from. Our gold standard assay for cell viability is the so-called cell titer glow assay. This is a bioluminescent lytic endpoint assay that quantifies the amount of ATP within your sample, and it also sticks to this admix and measure principle. So we have a single reagent containing all the required components. In this case, it contains all the components of the firefly luciferase reaction, except for a crucial cofactor, ATP. This ATP is provided by the sample, which are the cells, and which are lysed by the buffer of the cell titer glow assay. So this released ATP then drives the firefly luciferase reaction so that the light output and the amount of ATP can be correlated, and you can thereby conclude on the viability of your cells. An example for a cytotoxicity assay is the LDH glow assay. This assay indirectly determines the release or the loss of membrane integrity by determination of the amount of LDH released into the cell culture supernatant. And it does this by coupling the LDH reaction, so the oxidation of lactate to pyruvate, to another enzymatic reaction which converts a pro-substrate into the firefly luciferase substrate luciferin. So in the end, light output and the amount of LDH correlate. This assay is a non-lytic assay, and its bioluminescent nature makes it highly sensitive. And this is why you can use that assay also in a kinetic fashion. Only tiny amounts of sample are required, usually 2 to 5 microliters of the supernatant, which means you can collect sample at different time points, and you then have the option to either assay them directly or you collect all the samples and assay them together. Here's some representative data generated using that assay. This is with SKBR3 cells. That's a human HER2-positive breast carcinoma cell line that was treated with an antibody drug conjugate. And you nicely see the dose and time-dependent increase in the luminescent signal. Whenever you run an LDH release assay, you should always use heat-inactivated FCS, because the FCS is a considerable source of LDH, and you would thereby decrease the sensitivity of your assay by increasing the background. What you also should be always aware of is the fact that every cytotoxicity marker has its own time-dependent profile when it appears and disappears, and there are also markers which are really specific for a certain type of cell death. For example, while you can measure a caspase activation in apoptosis, you couldn't do this in necrotic cells. Or if we look at the release of LDH, this is an early event in necrotic cells, but rather a very late event in apoptotic cells, actually when these cells turn secondary necrotic. Or the decline in ATP levels occurs rapidly in necrosis, but more modestly in apoptotic cells. So you see the timing of your assay is really another important factor that you may want to consider. And this is further illustrated by that experiment where we treated cells with a cytotoxic compound and we assayed 3 different parameters: viability through ATP, we determined the activity of a so-called dead cell protease to estimate cytotoxicity and then also caspase activation. At an early time point, we see, as expected, a decrease in viability that is accompanied by an increase in cytotoxicity in caspase activation. However, the same experiment measured at a later time point, in fact, indicates a decrease in cytotoxicity in caspase activation. And this is exactly the problem that you may run into when using an endpoint assay and an activity-based biomarker such as an enzyme, because these biomarkers can undergo degradation or inactivation processes, and you could simply miss the time point that you actually would like to look at. A good way to approach this is to use real-time measurements, because then you can cover a larger time frame and you can easily identify the time point that you may want to investigate further with, for example, an endpoint assay in the future. I would like to give you two examples for such a real-time assay. One is the CellTox green cytotoxicity assay. This assay is based on a fluorescent DNA binding dye that is cell impermeable, so it's excluded from viable cells. However, once these cells lose their membrane integrity, it binds to DNA and yields a fluorescent signal. This assay supports the real-time course analysis up to 72 hours, so you could cover a time frame of 3 days. And also, you can nicely multiplex that assay with our bioluminescent assay portfolio. If you don't want to run a cytotox assay, but rather a viability assay in the real-time mode, the real-time glow empty cell viability assay is a good option. This assay is based on a purified luciferase that you would add to your cell culture supernatant along with a pro-substrate of that luciferase. In viable cells, this pro-substrate gets converted to the substrate, a process that does not occur in dead cells, so only viable cells will yield a luminescent signal. Similar to the CellTox screen assay, also this assay supports these real-time course analysis up to 72 hours. It is multiplexing compatible. And another advantage is that you can use the sample for downstream processing. So you could, for example, isolate RNA and run a real-time PCR. Besides the simplicity to identify correct time points with real-time assays, they also have an advantage with regard to assay costs. And this is exemplified here. So if we think of a real-time course analysis with a time point or data point recorded every hour, which means 72 data points, an endpoint assay would require us to use 72 microtiter plates and 72 times medium cells and reagents. And you can actually gather the same amount of data with a single plate, single times medium cells and reagents using a real-time assay. And with this, I'd like to hand over back to Jessica, who's going to tell you a little bit more about avoiding cryptic contamination.
Thank you, Erik. Okay, so there are, sorry, just have to change the slide. Here you go. There are two types of contamination I would like to talk about here because both are not so easy to detect and can have dramatic effects on your assays or any cell-based experiments that you perform. The first one is mycoplasma, a special type of bacteria which is too small to be seen in a standard bright field microscope. They don't cause any turbidity or color change of the culture medium like other bacteria do and are therefore often overlooked. The other type of contamination that often goes undetected are the so-called eukaryotic contaminations. These are cross-contaminated or even misidentified cell lines. There are estimations that 15 to 20% of all cell lines used worldwide are either cross-contaminated or even misidentified. I want to go a little bit deeper into the topic of how mycoplasma affects cellular metabolism by showing you some data from a study published in 2013. In this study, HeLa cells were infected with mycoplasma followed by analysis of the differential gene regulation upon different time points of infection. As you can see in this graph, the number of genes that respond to the mycoplasma infection increases dramatically with time. And this is just an extract of the list of genes that were deregulated upon infection. Let's come to the eukaryotic contaminations. Cross-contaminations of different cell lines usually result from accidents somewhere during the handling of cells. So it is basically a mistake in aseptic handling. Mislabeling of samples and inconsistent nomenclature of cell lines also contribute to this phenomenon. As soon as two cell types are mixed up, it is very hard to distinguish them. This process goes fast and it might go undetected, especially if the morphology of the cells is not drastically different. But although the phenomenon of cross-contaminated and misidentified cell lines has been recognized for half a century, as I mentioned before, still to date, we know that 15 to 20% of all cell lines used are either cross-contaminated or misidentified. I want to show you two examples of cell lines to give you an idea of the scientific as well as the financial implications once misidentified cell lines are used for studies. Due to a contamination in the 1960s, both of these cell lines are now known to be composed of HeLa cells, meaning scientists who thought they were working on HEP2 and IN407 were in fact likely experimenting on HeLa cells. It was calculated that over $700 million has been spent on published work involving just these two contaminated cell lines. And the amount of money spent on subsequent work based on those papers is even much higher. And these are just two examples that show you how that cell line authentication is a crucial thing. Okay, so before we start with our Q&A session, I hand over to Christian again.
Yeah, thank you, Jessica. Thank you, Erik, for this great presentation. I've recognized that we have a lot of people joining us today live. So I guess there will be a lot of questions for our Q&A session. But before we will start with the last poll question and the Q&A session, I promised you in the beginning that you will get a quick insight into some helpful solutions for your daily work, and I will show you in a second. So here are some product solutions that you might not be aware of. From Eppendorf, the first one is our CO2 incubator family Cell Expert that has been launched quite about a year ago. That might be interesting for you. So this is what it looks like, and I would just highlight some of the main benefits here. So it's very easy to clean. If you look inside the chamber, there's no fan. There's a deep drawn chamber inside, and one of the things that we've taken care of is a very high temperature homogeneity inside the incubator. So we measured at 27 spots inside the incubator the temperature homogeneity, so it doesn't really matter where you put your cell culture vessels inside the incubator. You always the cells always experience the same cell culture conditions. One thing that is also important in modern cell culture labs, especially for instruments that are running 24/7, are the running costs or costs that are related to internal parts that have to be exchanged over time, or especially gas consumption, which is a real important topic. And if you're interested in this, on how you can save money with the CO2 incubators, you can see on the left side of your screen. If you go to the resources section, you find a paper on cost of ownership for CO2 incubators, and also a paper on, or a little flyer on, how the CO2 incubator Cell Expert can help you to optimize your cell-based assay. All right, one thing that's also new are the 25 ml conical tubes that we offer, which you can see here. It's especially useful for improving contamination prevention in the cell culture lab, because you all know that if you don't have steady hands on Monday morning, it's really hard to pipette into 15 ml or 50 ml conicals without touching the rim or the rim of the vessels inside. So it helps you a lot if you have those tiny 25 ml tubes to prevent the contamination. And if you want to learn more, and even if you want to request a free sample, just go to eppendorf.com/25ml. One last thing I've mentioned before is our newsletter, Inside Cell Culture, it's called. If you want to get regular information for cell culture professionals, you get tips and tricks for your daily work. videos, downloads, posters, and more. You also get access to advanced educational webinars. And also, if you want to stay updated on upcoming webinars, just sign in, go to eppendorf.com/ICC and just type in your name and your email address, and you're ready to go. All right, so go one slide further. If you so let's come to the last poll question here. So we can improve our webinars for you. So the question is, how do you rate the content of today's webinar? We highly appreciate your feedback here. And we go directly to the live Q&A section of this webinar. So if you have any question that you would like to ask Erik or Jessica, please do so now. Just click on the Ask a Question box located on the far left of your screen. And we will answer as many of the questions as we have time for. So let's get started. Just choose a question. Let's have a look at this. So one question that we have is, let's see, I think this one will be good for Jessica. We use a multipad, not a multi-channel pipette, for seeding cells. Do you have any tips for using that to get the same amount of cells in wells? Jessica, this one for you, I think.
Good question. Yeah, so we check different, let's say, pipetting tools and how they can be used for seeding cells. And with a multipad, which is basically a dispenser, there is no need to resuspend or to swirl the cells up in between as long as you're fast enough. So you can be very fast with pipetting or with a dispenser and seeding out cells, and therefore you don't need to mix the cells like you would do with a multi-channel pipette and a reagent reservoir, or even a single channel pipette and the cell suspension in the tube. So with a dispenser, of course, it needs a little bit of practice with the handling, but with a dispenser you can easily fill a 96 well plate completely and have an equal number of cells in each well.
Yeah, thank you very much. Let's move on. There's an interesting one that's probably directed towards Erik regarding cytotoxicity markers. And the question is, regarding the LDH release, how long can I store the aliquots from the cell culture supernatant? Erik.
Yeah, thank you for that question. Here it is important that you dilute your sample in a storage buffer that is mentioned in the technical manual before you freeze them, because this buffer helps to preserve the activity of LDH. What we've tested so far is to store samples up to 1 month at -20, and we did not note any decrease in the activity of LDH or any change in the EC50 values of compounds that we treated the cells with. And I can imagine that probably also you can store them for longer times, but the 1 month is the time frame that we've tested.
Oh, thank you very much. So there are a lot of questions coming in. Wow. Let's choose another one. So this sounds also interesting. And the question is, how long does it take for cells to sediment in a reagent reservoir after mixing the cell suspension? 1 minute? Less? Is that the time available for seeding a plate before mixing again? And the question is, how do you mix a cell suspension in a reagent reservoir, pipetting up and down, shaking in an 8 formation? Who wants to take this one?
Yes, so maybe first, how do you mix the cell suspension? So I would recommend to simply pipette up and down to mix the cells, because shaking the reservoir always has the risk that you have splashes, and you don't want to have that under the bench. So when you resuspend, so when you pipette up and down, what a lot of people do is they simply overdo it. So pipetting up and down maybe 3 to 4 times is completely sufficient to mix the cells, and you don't have to do it like 10 times, because then you have the shear forces again that might stress the cells. And then the question, how long does it take for the cells to sediment? That is a little bit difficult to answer, because that will depend on the volume and on the cell number in the cell suspension. So you can say it maybe takes a few minutes until you would see it with the naked eye if you have a clear reservoir, but the cells already start to sink towards the bottom before. So I would recommend for a 96 well plate, resuspend 2 to 3 times every second row that you are pipetting, and then you then you are in a good range of a good mixing frequency. If you are not sure if that is sufficient for your cell concentration, then you can just try it out and count the number of cells that you seeded in the first row and compare it with the number of cells in the last row. So then you can be sure if the cell number is homogeneous over the whole plate.
Yeah, thank you, Jessica. I hope this answers this long question. Here comes a shorter question that is dedicated to Erik. So the question is, are the assays that you have been showing us before also applicable to 3D culture, Erik?
Well, primarily these assays have been designed for 2D culture. The non-lytic assays that I showed, such as the LDH glow assay or that real-time viability assay, they can be used with 3D cultures. We've used them successfully for 3D models like spheroids. If it's a lytic assay, such as the cell Tita glow assay, then of course the limiting factor for using them with 3D cultures is the lysis condition, because you have to achieve a thorough lysis of a spheroid, for example. So you have to make sure that the assay is achieving a thorough lysis of your sample. With regard to the cell Tita glow assay, we have a version that is optimized for 3D called cell Tita glow 3D assay. Here, the lysis strength and the protocol for that assays have been optimized. But in general, every lytic assay could be applicable to 3D cultures. You just have to make sure that the lysis is thorough.
Yeah, thank you, Erik. I think this answers the question. Let's go to the next one. I have one that's probably going to Jessica's direction. The question is, our incubator has a fan inside, so how can I protect the cells from vibrations caused by the fan?
Well, basically, you cannot, when you have a fan inside the incubator, it cannot be avoided that the fan is switched on. It is usually, I think it's dependent on which exact device you have, but it's usually switched on after a door opening, so and also in between when the door is closed, so you cannot avoid that. So depending on where the fan is located inside the chamber, I would just try to put my cells as far away as possible from the location of that fan to reduce the vibration as much as possible. But when the incubator has a fan, there is no way to avoid that the cells will be affected in some way by this vibration.
Yeah, thank you very much. I think it's also important to monitor your cells and see if there's any effect on the location inside the incubator, if you see any differences there. So the next question that we have is, do the substances you use in the real-time assays have any toxic effect on the cells? I've heard that a lot before, also in forum, and I think this one goes to Erik.
Yeah, I think so too. No, they do not. Well, at least for the time frame that we've tested, so the 72 hours, We didn't see any toxic effect. The way we checked for this is that we pre-incubated cells for that given time frame with the assay reagents, and we then used these cells in a subsequent assay and compared these IC50 values that we, for example, determined with a toxic compound with non-pre-treated cells, and we did not see any difference. So at least for that time frame, it's not toxic, but I'm pretty sure you could expand that also you could incubate your cells also for longer time frames. But that's the time window we tested because we think the 72 hours is also a time window that you would usually run an assay for, because if you think of how many cells you would have after 3 days, it's probably not applicable to run an assay longer than 72 hours.
Yeah, and I think if you do, you just use the same control as you do to be sure that there are no toxic effects if you run an assay longer than 72 hours.
Right.
Yeah. So let's go to the next question. We have a lot of questions still coming in. Let's choose this one here. How to detect eukaryotic contamination, how to avoid it is a question from Laurent. Who wants to take this one, Jessica?
Yes. Well, it's difficult to detect eukaryotic contamination just by looking at the cells because, as I mentioned, unless the cell morphology of the two cross-contaminated cell lines is not drastically different, you can hardly see that there's a cell line contaminating your cells. So whenever the cells suddenly look different than you're used to, then I would pay attention. But what you can actually do to really detect the eukaryotic contamination is an STR analysis. So STR stands for short tandem repeats, and this is the gold standard method to detect cross-contamination or to authenticate a cell line. And the question how to avoid eukaryotic contamination, there's a few things that where you can pay attention to. So one would be to have separate media bottles for each cell line and only work with one cell line at a time in the biosafety cabinet. Disinfect the workspace in between switching the cell lines and never use any reagents or any media bottles, aliquots or whatever for two different cell lines. So keep it as separate as possible.
Yeah, Jessica, thank you very much. I think we're a little bit over time, but there are so many questions, and we think you're still out there. We carry on for maybe another 2 minutes. So another question that we have is probably going to Erik's direction. The question is, can I only perform real-time measurements if my reader instrument is equipped with an atmospheric control? Erik?
No, it is also possible if you don't have an AC. The minimal requirement is really that you have a heating function in order to maintain that constant temperature. If you do not have a CO2 supply, what you need to do is you have to switch your cell culture medium from a bicarbonate buffer to a CO2 independent medium. These media use a different buffer, usually that's HEPAs, because otherwise your pH of the medium will simply increase over time, so it will become basic or turn pink, as I mentioned before.
Yeah. So thank you, Erik. I think this next question will be the last question for today. We will answer all of your questions afterwards and email you. But now let's go to the last question that is coming from Petra from the University of Ghent. And the question is, how do you defrost cells from -80? By applying heat shock for a few seconds or with warm medium? Who wants to take this one?
Yes, thank you for that question. So first of all, you should never store cells for a long time or store eukaryotic cells for a long time at -80. So the long-term storage temperature for eukaryotic cells should always be at least -150. -80 should not be used for longer than a few days. So I would recommend to thaw the cells as quickly as possible, because you usually use a cryoprotectant when you freeze the cells. Most commonly is DMSO, and this is cytotoxic when in non-frozen state. So you want to make sure that you get rid of the DMSO or dilute it as soon as possible. So you can, if you do a heat shock, you of course have to check whether this heat shock harms the cells. So just do a viability check, maybe 48 hours after thawing, so I would just dilute the cells with pre-warmed medium. And then it depends a little bit on the cell type you're working with. You can either centrifuge the cells right after thawing to get rid of the DMSO immediately, or you can dilute the cells and change the medium the next day. So for some cell lines are more sensitive to even low concentration of DMSO, and some are more sensitive to the centrifugation step. So if you don't know what is the method of choice for your cell line, I would simply compare both methods and then see how the cells behave.
Yeah, thank you, Jessica. Thank you also, Erik, for answering this question. This was quite a fast ride through the Q&A session. So we have received over 200 questions that will all be answered. So thank you again, Jessica and Erik. Do you have any final comments for our audience?
Thank you all for joining our webinar today. And we, as Christian said, we will follow up any questions via email and within the next days. So goodbye from my side, and take care.
Yeah, also I would like to thank you for joining the webinar. If you have any questions popping up later on, you can also contact us via email. Stay healthy, and thank you.
So before you go, I would like to thank you all for joining us today and for your interesting questions. As we said, questions where we did not have the time for today, and those that were submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the end of the registration. I would like to thank Jessica and Erik again for their time today and for their brilliant presentation, for sharing their experience. We would also like to thank Chrissy Jewell and the LabRoots team for supporting us today. And I'd like to make sure that you know that this webcast can be viewed on demand. And LabRoots will alert you via email when it's available for replay. And we encourage you all to share this email with your colleagues who may have missed the webinar today. Keep in mind to have a look at the Inside Cell Culture newsletter to stay updated on all upcoming cell culture webinars. So we say goodbye from Hamburg. Have a nice day, and most importantly, stay happy, stay healthy, and help to protect others as well. Bye-bye.